everyone, my name is Osamu Ibrahim. I'm an admin of the East Research Foundation, and I'd like to welcome you all to our inaugural webinar, which is part of our collaboration with Chen in Africa. We're going to be organizing a series of webinars related to science, so hopefully check them out as they come. Trend is a non-profit organization dedicated to fostering science education and research in the African continent and run entirely by volunteer scientists from different universities and research institutions around the world. We highly appreciate their contribution to the initiative, particularly Lucia Prieto and Tom Baden, and we look forward to a long and fruitful collaboration. Our webinar today, our first speaker is going to be Nobel Laureate Professor Martin Chalfie. Professor Chalfie received the Nobel Prize in 2008 along with two other fellow scientists for their discovery and development of green fluorescent proteins, or GFPs for short, which we will be talking about today. Professor Chalfie is also a professor at the University of Columbia. Also in our panel today is going to be our moderator, Professor Montasser Ibrahim. Professor Ibrahim is a professor at the University of Khartoum, where he works at the Institute of Endemic Diseases and is concerned generally with obviously endemic diseases such as malaria, and they do extensive research in molecular biology there. And again, we have Dr. Anwar Lafalla, who is the founder of the Sudanese Research Initiative Facebook page, who is going to be in the panel, as well as Dr. Sana, also SRF admin, Dr. Musab Al-Karim, who organized or basically made sure this event happened at all. It's through his work that we managed to set up this collaboration. Dr. Musab is a lecturer at the University of Nilain here in Sudan. So I guess without any further delay, the webinar will be starting with Professor Montasser Ibrahim as he takes over as moderator of the webinar.
Hello everybody. I'd like to Professor Chalfie, the presenter of this webinar. And although it's a virtual welcome in the sense that we have you here in Sudan, but we look forward to having you around. I think Sudan is an interesting place. It has a long history, and it has a history of science, and where a lot of things happened in the past. So we really look forward to conditions where people can travel freely to this country. I'm happy today that you are addressing those young science enthusiasts who look for role models in science. I believe one of them. Those young people are all full of hopes. We look forward to glow in science, but let's hear you on how organisms glow and what is the basis of that. Thank you.
So, Professor Martin, your mic is muted. Can you please unmute?
Trying to unmute it. Can you now hear me? Wonderful.
Yes, right. Please go ahead.
So I want to. I'm very honored to be asked to give this talk, to be the first in this series. I want to thank first the people at Trend and the people at the Sudanese Research Foundation for giving me an opportunity to speak. I do wish we could be doing this face-to-face. It's much more enjoyable, but this meant I could actually sleep in my own bed last night, and there's always something nice about that. So I greatly appreciate this opportunity. What I'm going to talk about is somewhat general about science and about how I think science is actually done. When I was growing up, let me start the slides, and we will. I can start the talk, but please, someone tell me that you can see the slides. You can, I hope. Well,
yes, we can.
Wonderful, thank you. So the story I'm going to tell is one that if I had thought about this when I was first thinking of being a scientist, that is when I was in elementary and high school, it has none of the features of what I thought science really was. Someone mentioned a moment ago about role models. I'm not sure there's any role model in this story, but people can take it for what they want. So what did I think when I was in high school? Well, I had a lot of strange ideas, and the ideas came to me because I would hear stories about the great scientists in the past. And hearing all those stories of the great scientists in the past gave me an image of what it was to be a scientist. And what I'm going to tell you in the story of the research I was involved in is that every single one of those ideas were wrong. So what were those ideas? Well, what did I, okay, I'm hoping that we can see the slides. Here we go. So what was I taught in the 1960s when I went to school? Well, the first thing was that scientists were geniuses. And if you weren't a genius, you shouldn't even start to do science, because that's all we heard about were these unbelievably remarkable people. And that's complete nonsense. The second thing about these geniuses, that it was something innate about them. It was something they had. They didn't have to do any work. It just came to them. And that meant if it didn't come to me instantly, I must not be a person that should become a scientist. The second thing was that all these stories that we heard of the great scientists of the past, Einstein, Darwin, Fleming, Newton, Galileo, all of these people, their experiments seemed to work all the time. So if you did an experiment and it didn't work, you were in trouble. The third thing that I thought these stories told me was that scientists use something called the scientific method. They're very purposeful in the way they do their experiments. So that the general idea is there's a problem. The scientist realizes that there is a problem to be solved, comes up with a hypothesis, and then imagines or figures out the very best experiment that will test the hypothesis. And the experiment works the first time because they're geniuses, and everything is wonderful after that. The third thing was that, or the fourth thing was that every story I heard about great scientists in the past, until people started in the middle 1960s to talk about Watson and Crick, is that every scientist worked alone. Now, for some reason, they could always have an assistant whose name was usually Igor, but other than that, they were loners. They didn't talk with other people. They didn't do anything with other people. They always worked alone. And the final thing about hearing about Newton and Galileo and Watt and Fleming and all these other scientists is that I got this impression, although I probably never said it in the words, that all scientists, except for Marie Curie and George Washington Carver, were white men. In fact, if you go on Google today and you look for famous scientists, what you get is the following picture. And there's a remarkable thing about this picture. First of all, everyone on here except Jim Watson is dead. And in fact, there's been studies in the United States that have said, can you name a living scientist? And most people cannot. But the other thing about this is that in this picture, they're all European or of European descent, and all of them are men, except for these two people, Marie Curie and Rosalind Franklin. George Washington Carver is even further down on the list. But this has nothing to do with what science is actually, how it actually is conducted now. And I want to show you a picture of the people that have been in my lab over the last 5 or so years. I've taken pictures of that to show you what a lab or really constitute, or what the people are that really are in the lab. And you can see here that there are people from Chile, Peru, Colombia, China, Iceland, the Gambia, Greece, and Israel, as well as people from the United States. So people that have come to my lab have been from all over the world. And in fact, this has been the case for a very long time in the labs here in the United States. That these are efforts that are really international. There's no particular country boundary here in doing science. So let me tell you now about the story of the green fluorescent protein and how I learned about it. Now, as I said, the usual story would be you should have a problem that you want to solve. Well, there are lots of problems, but I wasn't particularly interested in them, but I can present a couple of problems, 3 of them, in fact. I study the nervous system of a very tiny worm called Centauroidetus elegans. And so one of the questions is, can we actually see the cells that we would like to see? So how would we see particular cells? Now, the particular cells I study are cells that sense touch. And we know from diagrams of looking at the animal that these are very simple looking cells. I've drawn them here in green. But if you look in a living animal, we don't see that. We don't see any of those cells. So we would like to be able to see these cells in a living animal. The second problem is, if you look at an organism and study some part of it, let's say you're looking at cells dividing, how do you know what happens over time? Most of the procedures that people have used in the past have been procedures that have given you pictures, but you had, each one was a still picture. You had no way of looking over time, and you'd imagine how these might be put together. Sometimes it would work, sometimes it would not. Here, I guess we're not going to see that picture go. And then the final question that we were thinking about at the time is, how can we actually see that a gene is active? That the DNA is making RNA or the RNA is making protein. And we can try to do this in a number of different ways. So one way is we can look for the antibody. If we look look for the protein using an antibody, we can see where the protein is made. Or we can use the method of in situ hybridization, and that will tell us where the RNA is made. And finally, we can look using reporters that will turn on if the gene is active, and we can see those reporters. And we have been doing those experiments. So here is another picture of this worm that we were interested in. And we had been cloning genes, and we had been looking in the animal. And we could see, as in this slide, the red staining there, or the antibody staining, showed us that a protein that we were interested in was made in only a very few cells. Or we could use the enzyme beta-galactosidase. And again, if that was active, that was a reporter, and we could see exactly which cells that was made in. Or we could use in situ hybridization to look at the RNA. Now, the problems with this method, even though they gave us the answer, what cell makes what gene, the problem with these methods is that, one, it took a lot of work, and 2, the preparations were dead. We had to fix the animals and then permeabilize them so the antibody would get in, or the enzyme substrate would get in, or the probe for the in situ hybridization. So all we were able to get is a snapshot, not any idea about what was happening over time. And just look at that one instance. We wanted to look at a different time, we'd have to prepare animals for this. And every time we prepared this, every time we did the experiment, to repeat it, we had to do it all over again. Now, before I leave this slide, I want to point out something that's characteristic of all my slides. You see these two people on the right-hand side of the slide, Ongping Du and Shohei Matani. Their names are in red because these are people who have been in my laboratory. So anyone in my laboratory, that person's name will be in red. Anyone who's been a collaborator, their name will be in blue. And every once in a while, you'll see a picture of someone whose name is in black. That is a person who did something I wish I had done instead. So let me tell you how the real story is set up. So in 1989, I am working on this tiny worm, and we are interested in where genes are expressed. Now, I didn't say one other wonderful feature of this worm. This worm is transparent. When we look in the microscope, we can see these activities, the antibody and so on, because we can see right through the animal. It is a transparent animal. And in 1989, I had been telling people in my seminars that I worked on a transparent animal. These are the two key pieces of information that I brought to a seminar one day. I work on a transparent animal, and I want to know where genes are turned on. And so I heard the seminar by this man, Osamu Shimomura. And I'm not sure you can see the very bottom of the slide, but on that I've written nobelprize.org. And if you have a chance, I greatly encourage you to go and read his story. He is a remarkable individual. At the age of 16, he's told that he has to quit high school and work in a factory. So he leaves school, goes on the other side of the mountains that were on the side of the city he was living in, and worked in a paint factory. This turned out to actually be a very good thing for him because the city was Nagasaki, Japan, and it was 1945. So being on the other side of the mountains, he was protected when the atomic bomb destroyed the city. After World War II, he was able to go back to school, back to high school, finish that, but he couldn't go to college for several years because there was no college to go to. He finally did go to college, got a job as a technician, and he was given a project that we often give to people that, and we never tell them, and that is a project that other people have tried but have failed about. So the experiment has been a failure. We're hoping that the new kid will be able to do the experiment. In this case, Shimomura was able to do the experiment, and he was able, the experiment was a fascinating biological problem. How is it that a small crustacean called Cyprinina actually generated light? And we know that many organisms can actually generate light. Fireflies, glowworms, all sorts of insects, fish, fungi, bacteria, all sorts of different organisms, jellyfish, as I'll tell you in a moment. But they do this very differently from one another. So if you understand the chemistry of one, the biochemistry of one, you really don't have a clue as to the biochemistry of something else. And so he was studying how Cyprinina generated light. And he was able to get that to work. This led to two things happening. The first, he was invited to go to the United States to work on other bioluminescent organisms. And the second thing is he got a very unusual going away present from the professor that he worked with. The professor arranged for him to be given his PhD. Comes to the United States and decides he wants to understand why this jellyfish, Aquaria victoria, produces green light. and he starts doing the biochemistry. And as he does the biochemistry, he's a very good biochemist, nothing works. And nothing works for an entire summer. He's trying every way he can imagine to purify the protein that generates light, and he's getting nowhere. One night, he's working late into the evening, and the experiment has failed once again. He decides that's it for the day, and he cleans up after himself, and he throws the preparation into the sink. The sink also had probably some jellyfish parts in it, but it also had seawater from the overflow tanks that they were keeping some of the organisms in. He turns off the light and he's about to leave, and he happens to look back at the sink. And when he does, he sees that the sink is glowing. And he's very surprised at this because the samples had never worked before. And he goes back, he looks at the sink, and he thinks about it, and he says, you know, seawater has calcium in it. I've never tried calcium. Maybe that's why I'm suddenly getting a glow from this solution. And so he goes back in the next couple of days, and he does the prep again, and sure enough, every time he adds calcium, he's able to get a flash of light. And so he has solved the problem he wanted to solve. I have to say, this is sort of a rather good example of the scientific method, because you notice that the major technique here was throwing the sample in the sink. I've heard of people having great success throwing it on the floor, on the lab bench. It doesn't always work, but every once in a while, there's something unusual happens. In his case, it was throwing it in the sink. And so he used the ability that calcium could trigger the making of light to actually get to purify the protein. And he named the protein that produced light after the jellyfish. And he called this protein aquarin. I hope you can see that on the slide. But he had a problem. He had this wonderful protein that when you add calcium, it produced light, but the light was of the wrong color. The light that was produced was blue instead of green. And although he tried calcium on all his other fractions, nothing else would produce the green light. And so he thought about that and decided that maybe that there was a second protein, and that this protein would take the energy or the light produced by aquarin and convert that blue light to green. And so he took a handheld ultraviolet lamp and he looked at all his other samples, and sure enough, he found one that was green when blue light was shown on it. In 1962, he writes this up, a paper that actually is based on two accidental discoveries. The first, that required him to throw the sample in the sink, and the second one, that the protein he had was the wrong color. And so in that paper, which is mainly about talking about aquarin, he has a footnote that says, by the way, there's this other protein, and he called it the green protein, and he described its work. In that footnote, we, of course, now call it GFP, and aquarin, calcium, and GFP will now generate green light. And I believe this is the only footnote that has ever earned its writer a Nobel Prize. So you don't have to write a lot. Footnote is perfectly fine. In any case, as I heard the seminar speaker, a man named Paul Brehm, talk about Shimomura's work, I was fascinated because you'll remember, I work on a transparent animal and I want to know where genes are expressed. And I realized that if we simply took this protein and put it in the cells we were interested in, we would be able to see where it was simply by shining blue light on it. And we would see where it was green. And I got very excited about this. And although it's taken me a little while to tell the story, the introduction was much shorter than this. And I don't remember anything else in the seminar because I simply was fantasizing about all the things we would do, because we could use GFP, the green fluorescent protein, as a lantern to look at various cells. And so we started to think about how we would do the experiment. And in doing this, I collaborated with Douglas Prasher, who was in the process of cloning the gene. And I had a wonderful graduate student, Gia Skirkin, who just had come to the lab, and I told her about the project, and she started to do the experiment. We got the DNA from Douglas. She put it into E. coli, and 1 month after entering the lab, Gia had bacteria, as you can see on this page on the right, showing a picture of those bacteria that very first day that were strongly fluorescent. This was a very exciting finding. We were able to just put the gene into bacteria, then we knew we would be able to put it in anything, and we would be able to see where that protein was by shining light on it. Now, her lab page here, I've circled the part where she had strongly fluorescing E. coli, but I want to point out another part of this slide, and that is that she did the experiment not in my laboratory, but in a previous lab that she had worked in the engineering department at Columbia. And the reason for this is she tried to look at the bacteria under our microscope, but the microscope was a piece of junk and she just really couldn't see what she wanted to see. But she knew where a good microscope was, and so she went to use that. But you can also understand that this presented us with a real problem. We wanted to do the experiments, we had a wonderful result, but unfortunately we had no microscope to really use. So what I did was I simply called all of the people that sold microscopes in New York City, and I told them that we had developed a new way to look at where genes were expressed that used a fluorescent microscope, that we wanted to buy a new microscope, but we actually didn't have any idea where we wanted to buy it from. And so we had to sample the microscopes. We had to test them out. And so that we were able to do that. And I said to them, you know, if you leave it for 2 months or so, I think we can give it a good workout. And so we did all the experiments on borrowed microscopes. But fortunately, they worked. And after a couple of years, we submitted the paper. About 1.5 years, we submitted the paper for publication. And it was published. But before that, I want to tell you how unusual the experiment was. People that had worked with GFP before we did knew that GFP did not need anything extra added, but that GFP did something very strange. The molecule, I'm showing part of the molecule here, you see as it goes from serine 65 to tyrosine 66 and glycine 67, that's what's called the peptide backbone. And in all proteins, virtually all proteins, that backbone is completely straight. It's a linear array of atoms. But GFP does something very strange with 5 of the atoms, and that is it causes a cyclic compound to be made, this pentamere now that you see that connects this. This is absolutely needed for the fluorescence, but at the time, they knew this was needed, but they had no idea how it was made. They imagined that GFP needed to be activated by 1, 2, or maybe more enzymes. Now, Gia's experiment said, no, you don't need anything. It is its own activating enzyme. And that was a startling discovery. And that happened, it just happened. We gave it a try. We didn't know that it was going to work. In fact, people would have talked us out of it. Don't do that experiment. It's not going to work because you need the converting enzyme. Well, it turned out it did not need a converting enzyme. Interestingly, the way we did the experiment, and I won't go into the details of this, were slightly different than 3 other groups that I heard that was doing the experiment. And they did the experiment in a way that it would not work. And so, of course, they would then conclude, oh, there must be a converting enzyme. We did it in a slightly different way. We're very fortunate we picked that way. It was a sloppier way of doing the experiment, but it worked, and it worked the first time, and that told us we don't need anything else. So let me go from there. We did finally publish the paper in Science, and we wanted to publish in Science because it had the largest readership, and we realized that this way of marking cells and proteins was going to be a terrific way for everybody to, a terrific technique for everyone to use. And so we wanted it to reach as many people as possible. And that's why we chose the journal Science. This was not without some problems. Let me tell you a few of the problems. The first is that when we sent the paper in, the people at Science are very, very protective of what papers get published in their journal and which ones are not. And so they have an editorial board that decides not whether the paper is acceptable, but whether the paper is acceptable to be reviewed. And so I got a call from the editor, and the editor said to me, we sort of like your paper, but we're not sending it out for review. And I said, can you tell me why you're not sending it out? And the editor actually said, we don't like your title. I was a little surprised at this because I thought the title was very nice. Green fluorescent protein, a new marker for gene expression. But the editor informed me that I had used a word they never use in their titles. I had used the word new. And he said, everything we publish is new. You cannot use that word. So I asked him if that was possible for me to change the title. He said yes. And I think you can tell that I was not very happy that they were making me change the title, because the title I then submitted was a bit extensive. The title was, the Euphoria victoria green fluorescent protein needs no exogenously added component to produce a fluorescent product in prokaryotic and eukaryotic cells. This is basically the entire paper. I could have just sent in the title and the pictures and probably not anything else, but it was a very long time. But fortunately, the paper got accepted, and it went to press, and then the copy editor called me up and said, do you know that your title is quite long? And I said, yes, I do. And I said, would you like me to change it, shorten it? And the editor said, yes, please, if you could. And I said, well, how about this? Green fluorescent protein as a marker for gene expression. And everybody was happy. So that's the official title. So that was one problem. The second problem is, as you can see in the picture of the cover, we wanted to show, we wanted to have a cover picture because we had also put this not only into bacteria, but also into worms. And we were able to see this. And this is a picture I took of a newly hatched animal. This is an animal in which you can see the nerve cells actually growing out. And they're in the bright green of the GFP. And so I was very happy about this picture, sent it in. And then I got a call from the cover editor. And the cover editor said to me, we really like your picture and we want to use it on the cover, but we have a problem. The problem is that the only color that does not really reproduce well on our covers is green. Can we change the color? And I said, no way. And so this is actually a somewhat enhanced picture. If you really go to a library and see the cover, it is a little washed out, but I just couldn't have them make it red or purple or some other color here. So that this is the color as I saw it through the microscope. The third problem that we had in trying to publish this paper came about because we had already been giving away samples of the GFP gene to other people. And they were doing experiments in their labs, and they were getting back to us and say, you know, it works in our systems too. And so I wanted to say in the paper that GFP had been used in other places and it had been successful. And I wanted to point out these places: Dictyostelium, yeast, Drosophila, human cells in culture, that this is going to be a very general thing. And I wrote everyone asking them permission, and almost everybody wrote back and said, of course you can cite our unpublished work. You gave us the samples way before you published it, so of course you can do that. One person, however, gave me a very hard time. And this is her picture and the letter she had. It's a little hard to read the letter, so I've highlighted the most important parts. In order for me to cite her work, I had to make coffee. She asked that I make coffee every Saturday morning for 2 months, prepare a special French dinner for her, and I had to take out the garbage monthly or daily for a month. This is my wife. But what she did was a really wonderful, spectacular experiment. And you have to remember, genes have two parts to them. A gene has the part that encodes the RNA or the protein that's made, that encodes the product. But it also has a part that controls where, when, and how much of the product is made. What we had done in our initial experiments was to take just that controlling region, and instead of having it make the protein, it made GFP. So we could see where the gene was turned on, what cells turned on the gene. But the GFP would go everywhere in the cells. What my wife did, what Tula did, was to take both parts of the gene, the control part, the part that encoded the protein she was interested in fruit flies, and then add the coding region for GFP. And so now her protein would have GFP like that lantern hanging on it, so wherever the protein went, one would be able to see that. And I'm not sure you can see this in the pictures, but she works on fruit flies, and in the lower left part of the screen, I hope you can see that this is called an egg chamber. The cells on the left are called the nurse cells. The one on the right is the developing oocyte. And she was able to label a protein that was made in the nurse cells and actually see it go from the nurse cells into the oocyte and come to lie in very specific places within the cells. And so she was the first person to actually look at where proteins went using GFP. She claims that I still have not paid up all the things that I was supposed to do. This is something we debate about a lot. But it was actually wonderful to see that and see that result that she was able to get. So what are the advantages of GFP? First, it is encoded by DNA. So if you put that DNA into an organism, and there are many ways of putting DNA into bacteria or fruit flies or yeast or cells in culture or worms, once you've done that, then their progeny, subsequent cell divisions, subsequent progeny of the animals or plants, all have that DNA, and so you don't have to remake it all the time. You can establish a line that has GFP expressed in particular cells according to what DNA it's attached to. Having GFP in these organisms doesn't seem to hurt them. Now, if you overexpress any protein in any organism, that will cause problems. But having GFP on its own doesn't seem to hurt the organisms. And after all, you're just shining usually blue light on them to be able to see it. GFP is actually a very small molecule. It's only 238 amino acids, and a very compact one at that. And so it's able to go throughout the entire cell. And so cells with very unusual shapes can be seen. And I'll show you a picture of that in the next slide or so. And finally, GFP is a protein. Cells live with proteins. And so organisms that have GFP can be viewed when they are alive. They don't have to be fixed. They don't have to be permeabilized. All they have to be is the GFP is seen by showing blue light onto the organism. And so we can finally get not a static view of life, but a dynamic view of life. GFP has been used in lots of different organisms. To just talk about the cells, on the left side are worms, the ones we study, showing the entire nervous system of that worm. Fruit flies, and this rabbit at the bottom, this bunny is Alba, the GFP bunny. It's actually made by a French biotech company for the artist Eduardo Koch, who then would show Alba at his art shows to sort of get people talking about the connection between art and science. Alba lived to a ripe old age, as far as rabbits go, seemed to be perfectly happy, even though if you shone blue light on its skin, you would be able to see that it had GFP in it. In the middle, going from the top, are canola plants with GFP, some mice with GFP. At the bottom, I hope you can see a zebrafish. And on the right-hand side, you see a series of cells, and the one in the lower right-hand corner is a mouse Purkinje cell. And you can see how GFP has gone and diffused throughout the entire cell so that it's easy to see the entire cell outline. Now, I've said several times that this is something that one would, you can see cells dividing. So I'm going to show you two movies, or I hope to be able to show you two movies. We'll see if this works. And the first movie, the one on the left, has GFP attached to tubulin, which is part of the spindle. So it's only seen during nuclear divisions. Let me see if I can get that to run. Let's hope. No, unfortunately, the movie is not working. But one can go online to the American Society of Cell Biology and be able to see the cell division. This picture on the right, which now also won't work, unfortunately, is a picture of a series of nuclei. These are both in fruit flies. And here, GFP has been added to a small peptide called a nuclear localization signal. And that is brought into the nucleus whenever a nucleus is present. And so you can see in the middle, there's a nucleus before cell division and then two nuclei after cell division. And if I were able to show this film, you would see that at first, before the nuclei divide, the cells have all the GFP within them. Now, these are falsely colored, so it's still GFP, but the more that's there, the more you go towards the red end of the rainbow. So if you have a little, it's blue, a little bit more, it's green, a little bit more, it's yellow, and further to the orange and then red. So before the nuclei divide, only they have GFP in them. And then during cell division, when the nuclear envelope breaks down, it goes all over the cells to be absorbed once again into the animal. So I'm sorry that the slides didn't work this time, but I think these are available online. The third person that was involved in these experiments and shared the Nobel Prize with us was Roger Chen, who unfortunately died last year. Roger made many outstanding modifications with GFP. He made a version of GFP that was much more strongly fluorescent. He made other versions that were very useful. He was the first to come up with another color by mutating GFP to cause a different color to be made. He at first had a blue fluorescent protein and then a yellow fluorescent protein. And then after some Russian scientists discovered that corals had fluorescent proteins as well, one of which was red, he then took that and other similar molecules and actually generated a whole series of different colors. He wanted this for two reasons. The first reason was that by having different colors, you could look at different things, and I'll show you some pictures of that. But another was to take advantage of a very interesting property of fluorescent proteins, and that is that, or fluorescent molecules in general. A fluorescent molecule that is close together to another fluorescent molecule will, when they're appropriately matched, will not generate light, but transfer the energy to the other molecule. So he used this to make a very interesting method of detecting calcium. Because as you can see here, he took a blue fluorescent protein that was activated by ultraviolet light and gave blue light out. He connected that to a yellow fluorescent protein that would absorb blue light and give away yellow. But when there's no calcium around, these two molecules, or these two parts of the molecule, are far apart from one another. And so you get mainly blue and very little yellow. But if calcium is around, there's a rearrangement, putting the molecules very close to each other. And so now when the ultraviolet light activates the blue fluorescent protein, it makes very little blue light, but it transfers the energy to the yellow fluorescent protein, which now produces a lot of yellow. So by measuring the amount of blue and the amount of yellow, you actually can measure how much calcium is in the cell. And since this, like GFP originally, is entirely encoded in DNA, you can put this in any organism and actually watch changes in the calcium concentration within cells. And he and others have made hundreds of these molecular monitors. I mentioned before that he made these to be able to see different cells. I'll give you a couple of examples. In my lab, we studied this nerve cell that's right near muscle. And so we know that this nerve cell is, which we have GFP in, the muscle is RFP. When that muscle, or the nerve cell and the muscle are together in the newly hatched animal, in the larva, you can see that the GFP is right next to the red of the muscle because the nerve and the muscle are right together. But as the animal gets older, the muscle and nerve cells separate from another. It's because the surrounding skin actually surrounds and separates the nerve cell from the muscle. And so there's a gap between the green and the red. Well, we've been able to take many animals, give them a chemical to cause mutations, and look for adults that now don't look normally there because they don't have the gap. Now these adults have cells that are right next to the muscle. But probably the most striking use of multiple colors was done by Jeff Lichtman and Josh Saines at Harvard several years ago. What they were interested in is being able to label with different colors all the nerve cells in a mouse brain. And if you want to use all the colors of the rainbow to label mouse brain, there's only, I think, one term you can use for that, and that's what they called it. They called it BrainBOW. And you can see some of their pictures here, which are really striking as a way of following nerve cells separate from one another in a very complex structure that is the brain. I want to turn now to talk a little bit about how GFP has been used since we presented our paper in 1994. It's been estimated that in the 20 years to 2014, that GFP and other fluorescent proteins probably were used in over 160,000 scientific papers. And I like to divide examples of what they were into examples of fundamental science, applied science, and what I'm calling the unexpected. So for fundamental science, I'll give you one example from our lab. We really wanted to see our cells, and now you can see in this upper picture that our cells are nicely made fluorescent, and so we can look in a living animal and we can see these cells. But the minute you see the cells, you start to ask questions. Some of the questions are, why are there just 6 of these cells in the animal? Are there genes that control the number? Are there genes that control the position? Are there genes that control the outgrowth? And we have been able to mutate animals and simply look in the animals and find animals in which there are fewer cells, more cells, cells in the wrong place, cells with different number of extensions, cells that have the extensions go forward or backward rather than forward, all the different types of things that one could imagine. And then by identifying mutant animals that have these defects, we can then go back and study these problems in neurobiology. How are nerve cells made, and what are the controlling factors that make cells be different from one another? So that's an example of fundamental research. What about applied? Well, GFP has been used in many ways to do this. It's been used to study infection. There was a paper just a couple months ago, or within the last couple months, on looking how the mycobacterium that causes leprosy actually changes cells by looking at GFP expression. People have looked at how cancer cells metastasize, and they've also been able, as a movie that I'm not going to be able to show here again, how a cell infected, this is a mouse cell, infected with GFP, with GFP but labeled HIV, the AIDS virus, having that virus transferred from one cell into another cell, and by actually watching it. And it was somewhat unexpected because it's not a situation where the particle with the virus is just freely floating. It's actually transferred from one cell to another. And of course, that would mean that you have to really consider interrupting that transfer if you want to come up with a good way of controlling the disease. And so it's been used in many ways, applying it like that. It's also been used in biotechnology in a number of ways. But I want to tell you a little bit very quickly about some of the unexpected ideas. So one unusual use was something that was done by, it's an experiment that hasn't quite worked. And you will see why that's a problem. It had, because this is an experiment you'd like to work all the time. A man named Bob Berledge had the idea that he would take GFP, put it in a bacteria, but use the controlling part of the gene that controlling part could be turned on by the explosive TNT. And if that were the case, then whenever the bacteria were near that explosive, near TNT, and they were growing, they would now be able to glow green. So he tested this in a laboratory. He made the bacteria. He put them on a plate, a petri dish, with some of the explosive. And sure enough, when they grew up, the colonies were bright green. And then he was able to get 5 landmines. Now, he's not a stupid scientist, so they were disconnected, but they did have TNT within them. And he had a friend bury them in a 5 meter by 3 meter plot of land. And then he sprayed the area with GFP, and then he went back later to see at night where the green fluorescent bacteria were. And he was able to see that he could find where these landmines were hidden. Unfortunately, this does not work all the time. And I know of at least 2 other groups that have tried to work on this, and I suspect there are others that are still working on this. But of course, we want this to work all the time. False positives are okay, but false negatives are deadly. But this to me is a good example of someone who's thinking of an unusual application, using his brain to come up with ideas that could ultimately really help people. Because unfortunately, landmines affect non-combatants and are just horrible to have around. They're one of the worst aspects of war. So this would be a way of detecting it. On a somewhat lighter note, GFP has been used by some scientists in Japan. They've put it into silk moths, and they've actually been able to make fluorescent silk that they have then made into clothing. I'm not exactly sure what people would do with fluorescent clothing, because I think they would have to carry around ultraviolet lamps with them or blue lights to shine it on their clothes, but it's an interesting concept. And finally, people sometimes ask me, has GFP ever been put into humans? And this is not something that normally would be put into humans, but there was a hint that there may actually be an individual who really is the first human transgenic, and that's this individual here. The movie by Ang Lee, Hulk, has actually uses GFP as the explanation of why his skin turns green. Apparently, he's been injected, as you see in the very beginning of the movie, with some of the DNA from the jellyfish. It probably got into a gene that gets turned on when he's angry and is expressed in the green, in the skin. I actually know the screenwriter for this movie because he teaches here at Columbia, and I asked him how he knew about my research, and I was very proud that they used this in the movie. And then it turned out he had no idea what I was working on and didn't know that I was connected with this. He had heard it from a student who was working on the set with him when they were making the movie. So they decided to use the idea here. So there's all sorts of different things that one can use something for, most of which I would never have imagined. And that's one of the wonderful things about science, is that these discoveries get used by people to make other discoveries and to use them in ways that you don't even imagine are possible. So I want to finish by talking about what I think my research has taught me, some of the lessons here, because I hope you see that the story I've told puts a lie to almost every one of those things that were at the beginning that I talked about. So what do I think are the real lessons of this research? First of all, I think that scientific success comes in many different ways. I told you Shimomura's story of being told he couldn't finish high school, had to work in a factory, working as a technician, and eventually being able to make a rather astonishing discovery about a fundamental biological problem, how these jellyfish produce the particular green light that they did. Roger Chen has a completely different story. Roger's story is that he was really spectacular in science from a very early age. We have a contest here in the United States. It used to be called the Westinghouse Talent Search, Science Talent Search. And Roger won it when he was in high school. He was the best high school scientist of that year. It's rather remarkable. I have a completely different route to success, if you want to call it that. I did an experiment in college, or tried to do experiments, and I completely failed at them, and I took that as a sure sign of failure. And so when I graduated college, I decided I would never be a scientist. And I also had really horrible grades. I had C's, and especially in chemistry and physics. In fact, I often don't tell people about my grades because I somewhat fear that the Nobel Foundation may find out that I had bad chemistry grades and might take the prize away from me, but I'm not giving it back. But I was lucky enough, I got after college, I got a short job in a laboratory. And I found that among the other things that I had done wrong when I had first been working in laboratories is I never asked people questions. I never asked for help. And I discovered, to my great joy, that I was able to ask questions and I was able to think of new experiments. And every once in a while, those experiments work. And that's what seduced me into going into graduate school, because I loved the idea when the experiments work. So scientific success, there are no role models. You just have to enjoy what you're doing and get through the periods when things don't always work the way you want them to. The second thing is that I think many at least, if not most, discoveries are actually accidental. Shimomura was never trying to find a fluorescent protein, had never imagined that there were fluorescent proteins. But his accidental discovery of a quarin then led to his accidental discovery of GFP. If I hadn't been sitting in the seminar where somebody talked about this work, I would never have known about GFP, and I wouldn't have worked on that at all. So I think many, if not most, experiments are in fact accidental. I think that to do science, it is very good to be either ignorant, stubborn, or just have a willingness to try things. If we had listened to people before we did the GFP experiments, they would have talked us out of doing the experiment because it needed some sort of extra factor. Well, it didn't, and it worked on its own. But if we had listened to people, we would have absolutely not done it. Now, scientists often talk about what I would call a weekend experiment. This is an experiment you do Friday night, Saturday morning, Saturday night, Sunday morning, Sunday night, sometime on the weekend when you know no one else will be there. Because you just want to try the experiment. It's silly, it's stupid, it's not going to work, but you just want to try it. If it does work, and your idea was right, you feel very proud on Mondays and you tell people, oh, this is what I did over the weekend. If it doesn't work, all you just do is ask people how their weekend went and you just go on with your work. But doing those Saturday, Sunday experiments can sometimes be exceptionally rewarding. You have to try different things. Another way of putting it is you have to try lots of things in order for the accidents to happen so that you see the unusual aspects of it. I think that the stories we tell about science are stories of an individual and what he or she does, and we mark science by individuals. But I think that's completely wrong in this case. That scientific progress is really cumulative. It's not what Osamu Shimomura or Roger Chen or I did that made GFP important. It was the work of really thousands of people that gave it the impact that it did. It was a useful tool, but people used it in so many wonderful ways that it became a much more important tool, not because of what we did, but because of what everybody in the community did. In fact, I like to think about science very much like Roger Chen's monitoring with the two fluorescent proteins. You'll remember that those two proteins, the first one was activated, then gave its energy to the next one, and then a different light was given out. I think that's the same thing that we do as scientists. We take in results and observations from other people. We change them by our own experiments and thoughts and ideas and give them out again for someone else to pick them up and change them once again. And so you get this continual chain of interactions that actually make things much more important. Now, the next thing I'm going to say is going to sound like an advertisement. It's not really. But to me, what was essential was university and grant support. Columbia University doesn't care what I do at all, but they give me a lab to work in, and I have this complete freedom to work on any project I want to. I also have been supported by a grant from the National Institutes of Health in the United States. Now, they make me write a proposal. But again, it's not a contract. It's not that they're telling me what to do or that I have to promise to do something. People outside of science don't understand the difference between grants and contracts. Contracts are, I promise to do these 3 things, and when I do them, you will pay me. That's what business people do. They contract to get something done. And science is very different, certainly in biology. We say in our proposals, here are the 3 things we believe are the most important things to be done, and this is how we're going to do it. And if you agree with those 3 things, then please pay us, and we will start our experiments. But there's an unwritten part that says, you know, if someone discovers GFP or some other tool or CRISPR-Cas9 or something else that really helps our experiments, we're not going to blindly do what we wrote in our proposal. We're going to go off and use the new methods or the new ideas. Or if we make an accidental discovery and we think that's going to be more important, we're going to use that. And all the funding agencies completely agree with this, because why would you do an experiment that's 4 or 5 years old when you finally get a chance to go around it? You want to do the most modern things. So grant supports, as opposed to contracts, means that I have a freedom. As we did with GFP, there was never a proposal to do the work with GFP. We were working on other things, but it just seemed like the right experiment and an exciting experiment to do, so we did it. And afterwards, the NIH used it as an example of the types of science Congress should be supporting more. So grant support is essential, not a contract. The real people that are the ones that make the lab go are not the people that are the heads of the lab. It's the students and the postdocs in the lab that are really the innovators. In fact, when people started to learn that we had GFP working and they wanted to use it, the heads of the labs would call me up, and they almost invariably said the same thing. The first thing they said was, you know, my graduate student or my student or my postdoc has told me about GFP and they want it. What is it? And I'd explain it to them and they'd say, yes, that's right. Yes, we do want that. That's good. But it wasn't the heads of the lab that initiated it. It was the people working in the lab and doing things. And so they're really the innovators, and that's certainly the case in my lab as well. 2 more points. This story starts out talking about something, studying something that most people maybe wouldn't study. That is a jellyfish. But this jellyfish has given us several exceptionally useful tools. A quarrin first, bioluminescent marker, which was the first indicator of a rise in calcium in cells, and then GFP. And in fact, studying bioluminescence has been a terrific way of learning about all sorts of biology, not only by Shimomura, but by people like Woody Hastings and others. So to me, this says we should not be studying just a few organisms that are the designated organisms to study. We should be really studying all of life. And that will give us a much greater understanding of life. We often talk about the select group of organisms as the model organisms. I don't even like that term, and I'm sorry it's on my slide. But the reason is it's not modeling anything. These organisms, fruit flies, worms, bacteria, yeast, zebrafish, they're not models. They teach us new things that we had no idea about before. So I like to refer to these not as model organisms, but as pioneer organisms, because they're showing us something new all the time. And finally, this story started off looking at a fundamental biological process. How is it that organisms can generate light? That was the problem that he was studying. In my lab, we're studying how nerve cells develop and how they particularly function to sense mechanical signals like touch. But yet, these studies have allowed a wealth of applications in many, many different fields, including human health. So the final conclusion that I want to make is that fundamental research is essential. And I want to leave you or end this talk with my favorite quote about fundamental research. Now, this was said by the physicist Robert Wilson in 1969. In 1969, the United States was in the middle of what we call the Vietnam War. And so people were very much concerned about war, about defense, national security. And Wilson was asked to come to talk to the US Congress about the building of an enormous particle accelerator that Congress was going to have to pay for. And they were a little bit unsure about whether they would do it. It was built. It's called the Fermi Lab, and until the super collider in Europe was made, this was the biggest particle accelerator in the world. But in 1969, Wilson was asked to come and talk about the importance of this work. And one of the senators asked him, how is this going to help national security? And tell me how many ways. And Wilson gave him an answer he didn't expect. He said, none. So they asked him this a couple of times. Every time he said, no, it's not. Finally, the senator said, no, really, Dr. Wilson, in what respect will this particle accelerator help the national defense? At which point Wilson said the following to him. He said, it has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with whether we are good painters, good sculptors, great poets. I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. Thank you very much. So, I'm now back, and if there is time for questions, I'd be happy to answer them.
Thank you very much for very enjoyable and educating lecture or talk. Questions? But let me first start with the fun part of this. It's really enjoyable to look at all these experiments, you know, and I think fun is what, I mean, still working on science, again, is a low pay. I think we would never have of sort of doing science anymore. But can I ask you about the C. elegans? What was Leonard, another fellow Nobel laureate, and one of the few African or African-born Nobel laureates. Can you tell us about how did you end up using this, as you said, model organism? You know, you get it from Sidney Brenner, or?
So I was very fortunate. I went to England and I was Sidney Brenner's postdoc from 1977 to 1982. I went to the lab. I have to admit, I went to the lab for probably a very bad reason. Maybe I shouldn't admit this. I just thought it would be really nice to work in England. So I decided it sounded like exciting work, and as an added benefit, I could live in England for a while. So I thoroughly enjoyed myself doing that. But it was a wonderful opportunity to go and work in his lab. I had a friend who was already in his lab when I got there. This friend's name is Bob Horvitz, and Bob Horvitz is one of the three people that won the Nobel Prize in 2002. It was Sidney Brenner, Bob Horvitz, and John Sulston. So it was through Bob Horvitz that I learned about the work that was going on in Cambridge, England, and I liked it. I had some ideas about what I wanted to work on, and when I finally got there, I worked on something completely different that I had not expected, but it was the beginning of this work with these cells, these nerve cells that respond to touch.
Before I just give the floor to dudes, you know, to ask questions and other things, I'm sure they are can't wait to.
The dudes and the ladies. There's also ladies.
Excuse me. I just have one thing. You know, I'm focusing on the visit part, the whole thing. You know, would like to have you. As you said, physically here, but you know, one of the things that the Sudanese about 6,000 or 7,000 years ago have not, what do you say, discovered, but landed on, which is the fluorescence of tetracycline out of beer. You know, so if you come and go to the North Sudan and most of those cities, people found a lot of concentration of a tetracycline-like molecule, which fluoresces, as you know.
I see.
So you know, there are a lot of fascinating stories. You know, organisms opt for unusual solutions, and then it becomes part of the organism's way of survival and being unique. So what is the origin of all fluorescing stuff? Is it in the deep sea? Is it where? In the primordial soup? Can you just a bit?
So I'm not as up on all the work that people have done with fluorescence in different organisms, but I can tell you that fluorescence, at first, you know, people didn't really recognized that there were fluorescent proteins. I mean, it was the discovery that Shimomura had made in 1962 of GFP that was the first fluorescent protein, as far as I know. And they knew of other things that were fluorescent within organisms. But I think it was after our paper was published, that people started to look for other fluorescent proteins and find that they were very widely expressed. So people just weren't looking for them. So to get at the question of what is the origin of these, I'm not sure. As I said, I don't know about all the latest proteins that have been found. Some of them are quite different from GFP. But GFP itself is a very interesting protein. As I showed in one of the earlier slides, the protein actually folds up into a cylinder. And this so-called beta-can structure is highly unusual, but it's absolutely critical for its function as a fluorescent protein. There is another molecule that's an extracellular matrix molecule that's found in virtually all animals, I think, called nidogen, N-I-D-O-G-E-N, or maybe I-N. Nidogen has a very similar structure, and it's been estimated that that protein might just need 4 amino acids to be changed for it to be a fluorescent molecule. So it could very well be that this extracellular matrix molecule had developed over time, and then in some organisms, it had actually mutated to produce something that was fluorescent that was then acquired or became established within different organisms because it gave some advantage to them. So it may very well be that fluorescence could have evolved from a similar molecule, nidogen, in maybe many different phyla, very different species within a phylum, so that they've all derived maybe from one thing or one thing that's been modified. But the common backbone might actually have been there, or at least widely expressed throughout many organisms. However, I think some of the fluorescent molecules in bacteria are quite different from this. So fluorescence seems to have evolved in numerous times.
Thank you very much. Anwar, would you like to take, or guys would you like to come on?
Sure. I guess we will give a chance to the people in the call. So Anwar and Dr. Sana. I think Dr. Sana has a question because she's been moving her mic for a while now. So Dr. Sana, if you have any questions for the Chalfie, you can go ahead.
Yeah. Hello, can you hear me?
Yes, I can.
Okay. First of all, thank you so much for the very inspiring presentation. It was fabulous. You mentioned briefly the use of GFP and FRET or BRET sensors, and like wild-type GFP, like proteins, like tend to oligomerize or to form aggregates. And of course, like some monomeric forms have been constructed, but some of them might be incomplete monomer, which is a factor that we should consider when interpreting results. So, what are your thoughts regarding that? Thank you.
So, this was one of the lucky things that happened with GFP. GFP, on its own, expressed in cells, is usually there as a monomer. That's good. It doesn't need any other things. When the first red fluorescent proteins were found from corals, those proteins were obligate tetramers. And that's a problem, because if you have a tetramer, so if you have one of the problems, one of the proteins attached to another protein, as my wife had shown originally, then by making a tetramer, you're dragging the other proteins around. And it's known that if those other proteins have to make dimers or multimers of some sort, and at the same time you have, you need to make a tetramer, there's a problem with just everything precipitating within cells. And this had actually been seen with the previous marker, beta-galactosidase. That had caused some problems in cells because it also is an obligate tetramer. So this led to people doing a lot of experiments to try to mutate these other fluorescent colored proteins so that they only made monomers. That way they would not need to make any higher ordered structures, and so would presumably not interfere with whatever other protein they were attached to. So it is a very important aspect to this to make sure that they are the monomeric form. Usually all those colors that Roger Chen made are now named after fruits, so they're called M cherry for monomeric cherry colored or red, and so on.
Thank you.
Okay, thanks, Dr. Aslan, for the question. Dr. Anwar, do you have anything for Mr. Chalfie? Dr. Anwar, you with us? Your mic's muted if you're trying to talk. Okay. We still can't hear you. Can anyone hear Dr. Anwar?
No.
No. Yeah. Unless you were strong in your mic. So I guess I'll take a question from the floor and so this picks up. So we've had people watching online, like through our live YouTube channel, and I have at least one question from Japan. He is asking, will we be able to see GFP trees lighting the streets eco-friendly? So I think that's a bit of a broad question. I'm not sure how long ago, but to expand on it, because I'm a chemical engineer, and I'm generally interested in alternative energies and so on. So I would like to ask generally, is GFP viable as a source of luminescence? Like, what's the energy conversion like? Would we be able to use, for example, light from the sun and store it into biological material and have it light up in other times? Different places, different locations, different times.
So again, GFP is not bioluminescent, it's fluorescent. So it needs to have a source of input light or input energy to produce the light. There have been, every once in a while, after I first published the paper on GFP, I would get a telephone call from somebody saying, can we put GFP into plants so at night, if you drive by with your car, your headlights would turn it on and then you'd see green on the side of the road? And I think that that energy conversion would be very poor. GFP actually is a very good converting molecule. It has a very high efficiency. It's about 90%. That means for every 10 photons that come in of light, you get 9 photons out. That's an exceptionally high ratio. And so that's very good. But you'd still have to put some energy in. Now, you've asked this other question of, well, could there be something that could somehow store energy and then use that to activate GFP at night or some other fluorescent protein? I suppose it could. I'm not really sure of anyone that's doing anything like that.
Thanks, that was very helpful. We also have Shema, who is attending here. We have a kind of like a projector thing going on outside. She walked in and asked Shema Mohammed, medical biochemistry. She's a second degree in medical biochemistry. She's interested in neuropsychiatric diseases. And she was asking if we use GFP on a nano level or nanoscale, can we use it to conduct functional neuronal analysis and the connectivity of the neuron? So I have absolutely no background, and I'm not 100% sure how much sense this question makes, but
okay, let me see if I understand. Let me see if I understand the question. The question is, can GFP be used, or any of these sort of molecular monitors like the one I showed that Roger Chen made, can that be used as a way to monitor cellular activity, right? Or you measure things like nerve cell function and activity.
Yes.
And so the answer is absolutely. In fact, that calcium, and there's actually been a whole series of these calcium indicators. The first one, the one that Roger Chen made, was called Chameleon. And then a whole series of other ones were made based not so much on this transfer of energy, which is called FRET, but rather based on the idea that you can have two parts of a molecule. If they're apart, you get no fluorescence, but the calcium can then put them together, and once they've been put together, then they work. And so this has given rise to a whole series of what are called GCAMP molecules that have been more and more activated with different ranges of calcium, much more rapidly. And so we can see not the initial activation of nerve cells, but rather the consequent calcium increase that happens when a nerve cell is activated. Now, one of the holy grails of all of this is people would really like to see changes not in calcium levels, which we now can see, but rather changes in sodium levels, because that's the initial stage of many nerve cells that sodium rushes in to depolarize the cell. And so if there was a way of looking at that, and many people are still trying to get the perfect molecule to test that, and it hasn't, we're still not quite there, but there have been many wonderful things. Adam Cohen at Harvard is the ARCH molecules. There are other people, I think Udi Issakoff at Berkeley was really the first to try to do this, to try to take a molecule that had, for example, GFP on it, and by the change of sodium rushing into the cell might cause that molecule to be either more or less fluorescent. And so you could monitor the sodium transport into cells. We still don't have the perfect molecule for that. People are still working on it. But yes, these are molecular monitors, and they have been used to look at enzyme activity and kinase activity, protease activity, changes of pH within cells, changes in redox potential. All of these things can use these nano machines that are made using the fluorescent proteins.
Well, that sounds great. Thanks a lot for that answer as well. And I guess we'll just wrap things up and give the final question to Musab, who just logged into the call. I assume he logged in because he had a question. Musab, are you here?
Yeah, I can hear you well. Firstly, thank you so much for this terrific presentation. We really enjoyed it and gained a lot of new information. Actually, I have a question from one of the audience. He asked about, you mentioned in the last part of your presentation that basic research is very important, and we agree about that, but many African government and institutions give full priorities to applied research and neglecting basic research. Do you have any idea of how you can convince people to invest more in basic research?
You know, this is a general problem that is faced by everyone. We face it here in the United States. I've seen it in Europe, in South America. I've heard it from everyone that I've talked to. There is a fundamental wariness, I think, on the part of governments to support the basic research. I think my argument has always been that all the great advances that have been made in medicine and in engineering really started with basic research. We sometimes in the United States and elsewhere, they talk about what's called translational medicine. The idea that doctors now shouldn't do basic research, they should be translating what was learned in basic research into new cures and treatments. The problem I have with that, because frankly, as I get older, that's a wonderful thing. I'd like them to cure every disease. But the problem is that if you're going to do translational research, you must have something to translate. And I think that many non-scientists have a misguided understanding or a misunderstanding of how much we really know. There have been so many new discoveries that are made every year that enable many, many new ways of thinking about old problems. I mentioned in my talk that there is a paper on how leprosy might be spread in humans. Human disease, a horrible human disease. But the understanding in this paper, at least the hint of it is, that understanding what happens to stem cells, which is a very new area of science, is going to give us an understanding about how this disease is transmitted, or is how the body is affected by the disease. And it's a new perspective on this. There have been so many wonderful discoveries in the last few years, I would say 5 or 10 years, that have changed the way we do science. Just as I think I'm very happy that GFP has changed the way people have looked at a lot of different issues. And to give one other non-biological example, when Charles Townes, who was also here at Columbia University, did the first experiments in the 1950s on what became masers and then lasers, he was trying to address and think about some basic problems in physics. As those experiments succeeded and as more and more people started working with lasers, still in the early 1950s, no one came to him and said, I'm from the recording business. I think we can stop making vinyl records and we can make CDs and have our music on that. Or no one from the movie industry said, I want to put all these movies on DVDs. This will be great, and we'll use the laser for that. And no one from, in almost all the stores here, when you buy things at the grocery store, it's put through a scanner. Nobody said, oh, Dr. Townes, this is great, I'm going to use it for scanners. And nobody from the medical profession came to them and said, with this laser, we could do microsurgery, and we could do lots of different things with microsurgery. All those applications came later, and none of them were applications that he thought of or went into the work to do. But the basic physics that he was able to show gave rise not only to those applications, but to many other fundamental discoveries. The last one that I know of is last year, the discovery of gravitational waves that absolutely depended on lasers to be able to detect the presence of the gravitational waves. So my answer to this is that if you're going to have translational research, you have to have something to translate. And so scientific enterprises need both. They need people that are going to make the discoveries about things we know nothing about. And they're going to need people that are going to use those discoveries to make that applications that benefit all of us. So I think it's really a partnership, but I think that politicians don't like you to say, if we start studying the basic biology of this animal, it might give us some insights into some human disease maybe 10 or 15 or 20 years from now. Nobody likes to hear that. They like to hear, I'm going to work on a problem and I'm going to give you the answer next year. That doesn't actually happen in science. So it's science is a risky business. It is something that you cannot guarantee what the results are going to be, but the payoffs are enormous and I think very rewarding. I hope that's answered your question.
So I guess I'm finally able to join the seminar. So thank you very much, Professor Martin, for this insightful webinar about your research. I'm not from the biomedical background. I'm a computer scientist myself, but I'm interested in more in general about the policies and governance of research and education and so on. You have mentioned during your webinar, your presentation, that Columbia University just give you the grants. They don't.
No, the government gives me the grants. The university gives me the space.
Okay, okay, fair enough. So in the developing countries, especially in Africa, we have underfunding for research. So how can we overcome that? Any insights about this? Thank you.
That's a very difficult question for me to say. The first thing that pops into my head is that collaborations are really quite wonderful to make, and having collaborations with people all over the world is something that's done very regularly. And I don't know exactly how this is going to come out, but what I have noticed is that the education systems all over the world are really quite wonderful, and that no one place has any sort of lock or any sort of bragging rights of teaching their students better than any other place. We get spectacular, brilliant people from all over the world that I've seen in our department. So education is not, I think, that is not to me a problem. There's two other steps that I think are very necessary. One is an education step, and that requires quite an outlay. And the other is for these really accomplished people, each country must have ways of keeping them to allow their work in their countries. I mean, we often talk about a brain drain going from one place to another. And I think that governments, in my opinion, should really be thinking about promoting the scientists that they have and supplying them with the necessary reagents, supplies, support that are important. I'm not really sure how to talk anyone into doing this. I wish I had some sort of magic way of saying this. But I also often think that this middle part, the training part of scientists, not the education part, but the training of people getting their PhDs or postdocs, that can be distributed all over the world. But countries, I think, should support their researchers to be able to come back and to contribute within their own country. And then within that, I think this idea of collaborations is a very wonderful way of being able to do something more than any one person can do. This is something we do all the time. I think it's very important. There's many things I have no idea how to do, and having colleagues or associates that I can rely on is a very important way of doing science.
Okay, I guess that brings us to the conclusion of this webinar in particular. Someone just, I think that's Professor Muntasser, if you want to go ahead. And
that was a great pleasure. Thank you very much.
You're welcome. It's been a real pleasure being able to talk with you, even if it's through the computer. Maybe sometime in the future I'll be able to talk there face-to-face. That would be very nice.
Yeah, we've had multiple people asking actually if you would come and visit Sudan. But just to cut things short, we're just going to leave it up in the air as to whether or not you'll be able to come and visit Sudan. But again, we want to thank Chen a lot for getting us in touch with Professor Martin, and also we thank Professor Muntasser for moderating this session, and all of our panelists, Dr. Musa, Dr. Asna, Dr. Anwar. Of course, great thank you to Professor Martin himself for taking the time out to help us out with our inaugural seminar. Yeah, silent clap from Anwar over there. Yeah, so thanks a lot for that. I guess that's it for now. We hope to see everyone, all the audience, in our next webinar, whenever that will be. We'll keep you all posted. Thanks again, Professor Martin.
Thank you.
Anwar, you want to go offline? That would be fine. Or you want to just hang up? Okay, cool.
Okay.
Great, thanks a lot.
Yeah,
If you have any final words, you can go ahead.
Okay, goodbye.
See you.